So today I'm going to give you an overview on what we currently know about how bee health impacts honey activity. But before we can get to this, we first need to understand how honey impacts bee health. So first and foremost, honey is the main source of energy for the bee to carry out their day-to-day activities. Being able to store honey can also protect the hive from nectar shortages when foraging is not possible. And pretty incredibly, the capacity to store various products in the hive, so this includes honeys, resins, and pollen, is thought to allow bees to adapt their food source and behavior based on the health status of individuals or the needs of the colony, and this allows them to manage health challenges. Honey also plays a role in the detoxification of nectar during ripening. It can enhance bee longevity by preventing oxidative stress. It can influence developmental pathways and caste determination. And finally, it can promote cold tolerance in bees during overwintering. But most relevant to what we're going to be talking about today is honey's suite of antimicrobial mechanisms. So these can be broadly categorized into those that work when the honey is undiluted or those that work when it is diluted with water. So the undiluted mechanisms include high sugar, low water, and an acidic pH. And these all act to prevent microbial spoilage of honey during storage and prolong its life in the hive. The diluted mechanisms include hydrogen peroxide and compounds that are derived either from plants, from the bee, or from microbes. So we'll go into what these are in a little bit more detail in just a moment, but what you need to know for now is that ensuring protection of this diluted honey is essential to the hive. This is primarily because nurse bees need to dilute the honey with water before feeding it to larvae and adults because of its high viscosity. So here we have a table which summarizes results from all of the published studies out there that have tested the activity of honey against important honeybee pathogens. So this includes the bacteria that are responsible for American and European foul brood, the Microsporidium nosema, and the fungi responsible for stone brood. Unfortunately, there haven't been any studies published to date that have looked at chalk brood, but for the rest of these hive pathogens, you can see that quite a diverse variety of both monofloral and polyfloral honeys have some activity against them. So this includes a lot of honeys from native Australian plants, such as bottlebrush and jarrah as well. So here we've done a simple experiment looking at what microbes are able to survive in honey. So these pictures are of agar plates, which are full of nutrients for microbes to grow on. The two different colors are just different plates which either bacteria or fungi prefer to grow on. And what we've done is we've taken different honey samples, we've dipped a swab in them and spread it on the agar plate and then left them to grow for a couple of days at hive temperature, which is about 35 degrees. So can you guess which of these are active honeys and which are inactive? Well, if you guessed that the ones on the right were the active ones, then you are correct. So these results are pretty dramatic, and they're even more impressive when you consider that these honeys were actually collected between 2005 and 2007, making them around 15 years old. So this just goes to show the amazing power of active honey to prevent microbial growth and to protect itself and the hive from microbial attack. So now that we understand a little bit more about just how important honey is to bee and hive health, it's quite easy to see that the reverse could also be true, that bee health likely also impacts honey activity. So coming back to our diagram here of the antimicrobial components of honey, in the context of commercially active honey, we're now mostly interested in these components that work in diluted honey. So we're going to go through them one by one and talk about how they are potentially affected by bee health. So let's start with hydrogen peroxide production. So hydrogen peroxide is the main source of antimicrobial activity in most honeys. It destroys microbes by causing oxidation of their proteins and DNA. Hydrogen peroxide is produced by an enzyme called glucose oxidase, and this is added to honey from the salivary glands of bees during processing. Interestingly, high glucose oxidase activity is not clearly associated with any specific floral sources. So honey from any given floral source might have high, low, or no glucose oxidase activity. Instead, studies have found that bees that forage on a diverse diet of varied flora tend to have elevated levels of glucose oxidase. And this also appears to correspond to enhanced immune functions and better in-hive pathogen protection. So it looks like this is a feedback loop where healthy bees produce glucose oxidase, which produces lots of hydrogen peroxide in honey, which in turn protects the hive from disease and helps the bees to stay healthy. So here we have a table which shows the antimicrobial activities of 32 polyfloral honeys from New South Wales. So these were taken from a larger survey of several hundred honeys from all over Australia. The activity of these honeys was determined by the industry standard phenol equivalence assay, In the blue column, you can see the total activity, and in the green column, you can see non-peroxide activity. So the dashes here just mean that the value of the non-peroxide activity was 0, which shows us that the activity of these honeys was entirely peroxide-based. So these honeys had peroxide activity ranging from 8.1 to 29.8, with an average of 18.2. For some context, anything between 5 and 10 is considered low activity, between 10 and 20 is considered therapeutically beneficial, and above 20 is considered high activity. So in wound gel and ointments, honeys with activity 18 or above are generally used. So you can see that these mixed flora honeys are really quite active, with the average being 18.2. So this seems to hold true to what we would expect based on the previous information. So next we have a look at other bee-derived compounds in honey. So what are these compounds? Well, one major one is bee defensin. This is an antimicrobial peptide that is part of the bee immune system. And it has activity against a wide range of bacterial pathogens, including the bacteria responsible for European foul brood. There are several other antimicrobial peptides that are part of the bee immune system, but these are not as well understood as bee defensin. So we also have the major royal jelly proteins. So these are, as the name would suggest, found in royal jelly, but they also happen to be the most abundant protein in honey, and they break down into smaller antimicrobial peptides. So studies have shown that various phytochemicals that come from nectar and that are found in honey upregulate production of these bee-derived immune-related proteins and peptides. So again, it seems that healthy bees will be producing more of these active compounds that end up in the honey. Now we come to plant-derived compounds. So these are known as phytochemicals, and these encompass numerous diverse chemicals with various functions. So in this list here, you'll probably recognize MgO, which is the active compound in Manuka honey. But we also have an array of other things, including polyphenols, alkaloids, and flavonoids. So many of these phytochemicals have actually evolved in plants as a protection system against stresses, including microbial infection. So it's quite likely that they would perform a similar function in honey. Most of them, however, are found at concentrations that are not sufficient to account for significant antimicrobial activity on their own, and they are thought to work in combination with other compounds that are in the honey. So this means that it is perhaps not the quantity of any one thing, but the diversity of different things in the honey that can significantly contribute to its activity. And therefore, it is again likely that foraging on diverse flora leads to a diverse plant chemicals in the honey, which therefore enhance its activity. And finally, we come to bee health and the microbiota. So the microbiota here means a collection of microbes. This could be all the microbes which live in the nectar taken from plants, or all the microbes living in the bee gut, or all the microbes living in the honey itself. So currently, just how the microbiota of bees and honey changes with bee health and exposure to different floral sources is relatively unknown. Some recent studies have suggested that honey can contain a huge variety of these compounds, and some of them are likely to impact its activity. A few antimicrobial peptides that were produced by bacteria have been isolated from honey samples, and these have been tested and have shown activity against hive, human, and food spoilage pathogens, including both bacteria and fungi. So these microbes also likely play a significant role in the breakdown and digestion of other compounds in the honey, and this could either potentially enhance or neutralize honey's functional properties. But again, just how the microbiota of bees changes with bee health is relatively unknown. So now we've heard the story, where to from here? So the picture that we've been building up is one of a feedback loop between biodiverse forage and healthy hives, healthy bees and healthy microflora, and in turn, active compounds and active honey. So the flip side of this is degraded ecosystems and unhealthy hives, poor bee health and disrupted microflora, and in turn, less active compounds and inactive honey. So these observations move us away from the desire for monofloral honey produced from single plant species and plantations towards honey that is derived from diverse native floral sources. As part of the New South Wales Bushfire Recovery Project, we're currently collecting diverse samples of honey and bees from across New South Wales, and we're working on several projects in this area, including investigating the activity of hive products against hive pathogens, looking into the relationship between bee health and the bee gut microbiome, and more. We really hope to be able to fill in some of the questions which are still remaining in this picture, with the ultimate goals of helping beekeepers increase honey activity through improved management practices and promoting healthy native New South Wales forest and bushland. So thank you very much for listening. And if you're interested in reading more about our work, you can check out the website at the address shown on the slide.